Hi everyone, welcome to Ward Science webinar on the genetics of taste. My name is Asno Seep. I'm a lab technician here at Ward Science. Today we're talking about the genetics of taste, how phenotypes and genotypes correlate. All right, so today we're going to be looking at the genetics of taste, a kit provided to us by Geno Sensor Core. They've been great help to us so far, and so we're going to be looking at a quick DNA isolation of cheek cells, followed by a polymerase chain reaction, digestion, gel electrophoresis, all very basic standard science templates that we use when working with genetics. So one of the main issues with teaching genetics in the classroom is that there's a disconnect between a genotype and a phenotype. It's very easy to describe to a student what a phenotype is. My hair is brown, my eyes are blue. But once you try and bridge the gap to a genotype, you start running into a kind of, how do you see that? How do you test it? You can take someone's word that they have gene whatever, but with this kit, we're actually able to see not only a positive phenotype, but link a genotype directly to it. Now, to do that, we're going to be using these genetic tools I just spoke of. Now, safety is one of the big concerns of this lab. Not that there's anything particularly dangerous, anything we're doing, but keep in mind that safety and the tools we use to keep safe are just as important for us as they are for our reagents and our any samples we have. We don't want to contaminate anything. We don't want to accidentally add anything more to the reaction. So for this, we're going to be using eye protection because we are working with a couple liquids, gloves because we're going to be touching a lot of things. We don't want to transfer any chemicals we have on our hands to any of our reagents. And same thing with the lab coat. We want to keep everything as separate as possible. So we'll get started. I'm going to put on my gloves. Oh, it's stuck in there. And what we're going to start with is a quick isolation of cheek cells. So the cheek cells we have today are going to be provided by yours truly. And to do this, it's a very, very simple thing. We're going to start with a quick PTC paper test because we're going to be looking for a gene directly linked to the ability to taste PTC paper. Now, if you've never actually tasted PTC before, it is something that's really fun. It's included with the kit, so you can actually make a couple predictions on what you might be. The gene itself is split into 3 different genotypes. You're going to have your homozygous positive, meaning that not only can you taste the PTC, or I'm sorry, you can't taste the PTC paper. The homozygous wild type cannot taste PTC. The mutant allele can, however, and then people who have both alleles, meaning they're heterozygous, will be able to taste a little bit. Now, I have the GenoSensor Kit right here. This is all of the room temperature reagents. Things flying everywhere. And in this, I actually have some PTC paper. This is great because it is a very easy thing to test. You could pass this out almost like candy, but it's very easy, and a student can have this very quickly, very easy, right before you actually start anything. You pop it in and see if you could taste it bitter or not. For example, some people have a very strong reaction to it. It's very, very powerful. They can barely stand it. Some people can't taste it, and some people are in the middle. I'm in the middle somewhere. I can taste bitter, but I'm not too, I'm not overwhelmed by it. And what we'll do is we'll see that translate to a genotype down the road. So to actually collect the cells, we're going to start with a microcentrifuge tube. And I'm going to fill it with 200 microliters of genotype solution A. This will be collecting the sample. Now, solution A is a lysis solution, meaning that it's going to break apart the cells. Now, the DNA is caught up in the cells right now, and it's something that we're going to want to get at eventually. So to actually collect the cells, now that that's in there, I'm going to start chewing the inside of my mouth just a little bit to loosen up the cells. Chewing it inside your mouth, kind of working your teeth next to your gums, rubbing it against your teeth. This will loosen enough cells so you can get a pretty good, starting inoculum of your own cells. Now, when you do this, be sure that you haven't had any food in the recent past, because you don't want any bacteria or a huge amount of bacteria in this sample. You want it to be mostly your cells so we don't confuse any of the reagents, because we're going to be looking at finding a very specific DNA sequence down the road here. So,
excuse me, I have a question.
Oh, sure, of course.
Should I brush my teeth or use some mouthwash prior to?
Sure. No, just as you are. There's no need to brush your teeth, or I'm sorry, if anybody here, the question was, do you need to brush your teeth or use mouthwash before you actually isolate the cells? There's no need to do that. You're going to want to have as much of your own, cells available as possible. The only concern is don't, eating chunks of food. Don't have them laying around. You don't want to get those caught up in the reaction. But no need to brush your teeth or use any mouthwash. So I've chewed of course. I've chewed the inside of my mouth, so the cells are pretty loose. The inside of my mouth feels kind of raw. So what I'm going to do is I'm going to work around this cotton swab in my cheek for about 10 seconds. I'm going to want to get as much of it as possible. There we go. Oh. A little bit bonus. And right in. Now what we're going to do is use some scissors to cut the tip of that cotton swab right into the cell. Right into the tube. Now with the tube, we have our solution A, and we also have our sample. What this is going to do is we're actually going to use a handy-dandy heat block, which has been set to 95 degrees. Now this is going to help the solution break down those cells and get that DNA into the solution so that we can extract it later. Now, a handy thing about using tubes and a heat block is this will be set to 95, and I cannot tell you how many times I have burnt my fingers. I've seen people burn their fingers trying to get tubes out of here. The quick solution, an easy solution to that, is actually using a tube holder. Now, the tube holders are great because they fit right on. Not only do they prevent your fingertips from being burnt, but they also prevent the cap from being blown open because we are essentially boiling water in a tight space. So, in there for about 10 minutes. After we're done incubating, we'll have all that in solution. All the DNA will have come out, and we'll be able to actually use it. Going back, just rewinding a little bit, we've incubated this at about 95 degrees Celsius for 10 minutes on our heat block. Now, I was mentioning that it's really useful to use a microfuge holder, which is a small plastic insert you can put right on top of it. This is nice because it prevents you from burning your fingertips as well as keeping the tube shut so that it doesn't accidentally pop open, because you are boiling a bit of water, or near boiling, a bit of water at high temperature in a closed environment. So if you ever hear any popping noises coming from your lab, that's what it is. So by using a holder, you're able to prevent that. Now, after it's come out, what we'll do is we'll actually spin it down because we want to get as much of those cells off of the tube as possible. Now, I have a handy little microfuge which allows me to do pulses. Any microfuge will work just to pulse it down. When you put it in, be sure that you have a counterbalance to it because it makes a terrible sound if a microfuge is off balance. So pulse it for a couple seconds. That will get all the cells. There we go. I'll get all those cells off of the cotton swab into solution, and we'll be able to continue from there. What we're going to do now is we have to get that cotton swab out of the solution because we're going to be working with the rest of the chemicals. To do that, we're actually going to be using a sterile technique because, like I said again, we don't want to add anything new to the solution. I have ethanol and a little pair of forceps here, which I'm going to use to sterilely remove that cotton swab. And we're done with this. Let me get this out of the way. All right. Now, we're not too concerned about bacterial contamination because we're going to be working with the actual genes and the DNA of a solution. We're not going to worry too much about any bacteria getting in there and ruining things. That's not to say be willy-nilly and go crazy with, not wearing gloves or personal protection. but you are going to want to be aware that, you don't have to be super sterile. Now, I've removed the cotton swab, and what we're going to do now is we're going to add solution B, which is another room temperature solution. What this is going to do is it's going to nullify the lysing agent. It's going to turn everything back to being kind of a standard pH. It's going to remove all of the nasty bits. I have it right here. And only to do that, it's only about 20 microliters, so not too much.
Yeah, one quick question.
Mm-hmm.
They do not have a heat block. Would they be able to use a water bath or an incubator?
Yes, so long as that water. So the question was, if you don't have access to a heat block, can you use a water bath? The answer is yes. Anything you can use to get that tube to 95 degrees for 10 minutes will work. Be that a water bath, a heat block, or anything you can do. Would I recommend putting it in the oven? Not so much. But a water bath will work fine. Boiling water, perfect. So I'm going to add 20 microliters of solution B to this. Like I said, that will nullify solution A, and we'll be able to move on from there. Now, solution B, once it's added, there we go, goes away. Now, what we have here is a mixture of DNA, cells, cell walls, proteins, kind of a bunch of gunk you don't want. To get this out of solution, we're actually going to centrifuge this for about a minute at 12,000. Now, I don't have my big centrifuge here, but I'll throw it in here just to illustrate the fact that we're going to be doing that. Once you've centrifuged it for about a minute, you can take it out and you actually see the cheek cells on the bottom. Now, those aren't what you're looking for. The cheek cells, or at least the matter particulate you see on the bottom, those are the proteins, the cell wall, all that junk you don't really want. That's not going to show up well, and it's not going to tell you anything about if you taste that paper or not. What you do want, however, is going to be the supernatant, which is floating above it. Now, I'm sure you can't see it on there, but I do have a bit of particulate on here. Now, all the clear liquid above it is going to hold my genomic DNA. So, this is actually a good stopping point because you can freeze this at -20. So if you have a class or something, you can, isolate cells, throw it in the freezer, come back the next day. You'll be able to do that. So, quick tips on isolation, because it is while it's not a taxing endeavor, it is something you have to keep in mind when you do it. I did mention the tube holders. They're phenomenally helpful when it comes to working with it. It avoids burning your fingers. It's really nice. And as I said before about sterility, it's something that is recommended. You don't want to be adding anything random, any new bacteria. You don't want to be touching your samples too much. It's really about protecting those samples when I say sterility. And then finally, the PTC paper is a great tool when it comes to actually, predicting what your genes will be. So it's fun to sit down, taste it, see if you make some predictions, and then later on we'll see if those actually panned out.
Someone thanking you for that tip about freezing to save time during their class.
Yeah, so freezing is a very helpful tool we can use, and there's a few points in this experiment that you're able to segment this into different days. So right now we're going to go into the polymerase chain reaction. This is really a revolutionary tool, or at least it was a few years ago when it first got developed, because this allows us to take a very small amount of DNA and make a whole bunch of it. So what we have here is, remember, we're looking for the gene to see if you can taste that taste the PTC paper. That's a very specific gene, and there's probably not too much of that in here. If I were to run this on a gel and try and find it, odds are I wouldn't see anything. To get around that, I'm going to be using the polymerase chain reaction. To do this, we're going to need some PCR tubes, a thermal cycler, and a bucket of ice, just to keep everything kind of cool. Now, Geno Sensor is kind enough to provide not only positive and negative controls, so you can actually see what it's supposed to look like, but also a master mix. That master mix is going to contain things like the polymerase, primers, which are going to be identifying those gene sequences, as well as any buffer and nucleotides that we'll need to make a whole bunch of DNA. Now, the PCR of cheek cells is a very straightforward process. We're going to start with a very tiny PCR tube, and we're going to put about 2 microliters of our own DNA into it. Now, here we go. This is the one. Now, one of my, when I first started doing this was, it looks like there's not a lot of, 2 microliters is not that much. But keep in mind that PCR is a very site-specific process, and we're going to be making a whole lot of DNA, so there's really not too much of a need to start with too much off the bat. Now, when you take this DNA from the tube that you've already isolated from, be sure that you don't mess up or mess with that pile of cells on the bottom. That's all the stuff you don't want. Any proteins, any cell wall material, anything you find in there is going to kind of throw a wrench in the machinations of the rest of this. So 2 microliters of the DNA that we've recovered. That can go away. And then 8 microliters of RNase-free DH2O. Now, the RNase-free DH2O is actually provided by Geno Sensor. It is the negative control, which will also be running on our gel to make sure that there's nothing wrong with any of our solutions. So RNase-free DH2O, that'll go in. That gives us some room to actually allow that reaction to occur. Now, I'm doing these in this order, not because there's any particular reason, but because that's just the easiest way for me to show this. Now, what I'm going to do next is grab the PCR master mix itself, which I've been keeping on ice. Now, keep in mind that this is full of enzymes, protein bases, nucleotide bases, and other things that we don't really want sitting at room temperature. We don't want those to degrade. We don't want the DNA to, anything wrong to happen with that. So this whole thing, this whole process can be done on ice, and generally it's recommended you do it on ice so that you're able to maintain as much of that as much of that polymerase as possible, as well as you don't want to mess up any of the DNA. Now, I'm going to keep doing that. The polymerase itself, we're not using too much of it, only about 10 microliters. Now, one hand at a time. Now, this is good because it has all the stuff we need to make a whole bunch of DNA. Like I said, the polymerase mix will contain the polymerase itself and just about everything else we need. Now, we're going to be moving into the PCR itself very soon here. What you're going to want to do before you put it in there is mix this as much as you can with the pipetter. You really want to make sure that it's kind of a homogeneous solution because you don't want anything missed. It's going to need all the reagents, and we don't want to miss any of them. All right. Put that back. And I'll put this back on ice. So now that we've prepared the mix, we can actually move into running the PCR machine itself. All right. So the PCR machine I have back here is a Ward Science PCR machine. And these are very varied when it comes to how they're run and how they operate, how to program them. Generally, a PCR machine is going to run on 3 basic principles. It's going to have a step where it breaks down the cell or the DNA, which is the unwrapped DNA. Doing that allows the second step, which is annealing. Basically, those primers are going to bind to very site-specific regions of that DNA, meaning that we'll be able to actually find the genes we're looking for. The following step is the elongation of that chain, which is where the polymerase will then bind to those primers and then elongate. So we take one strand of DNA, break it apart into 2, add primers. Now we have 2 strands of DNA. That'll repeat itself for about 40 cycles or so. All in all, it takes about an hour for it to run. So if you're going to run it in a classroom, keep that in mind. This is actually a great opportunity to talk about some of the intricacies of DNA itself. How it's made, how it's replicated, and anything else you do with DNA. And there's a lot of fun stuff you can do with it. There's actually quite a story with PCR itself. If you ever had a chance to look up Carrie Mullis, I highly recommend it. Now, loading the machine itself is very straightforward. There's a little space in the middle of which all the tubes go, and you'll able to find just about any room you can and put them in there. Now, keep in mind, some of these machines have spring-loaded tops. The tops are really built so that the PCR tubes are pushed down and are snug as possible, because the thermal cycler is going to be going from all those different modes I was telling you about. So, the annealing, the breaking down of the DNA, and then the rebuilding of that DNA. And it's going to be going back and forth for a while. So those tubes are going to be in there real snug. Now, some machines might have a manual tightener, and just be aware of what machine you're using, because some of them are different, but they all operate on the same basic principles. So, like I said, this will run for about an hour. It's kind of out of your hands when you do that. So this is a great opportunity to talk about other things. Once the program is run, you'll have your tube, and this could also be stored at -20. So another great stopping point for further down the road, if you so chose. All right, now PCR, like I said, is a very, heavier than I thought. It's a very tried and true method. It's very common. A lot of people run it, and it's a great way to test, is DNA there? Can you find it? And if so, can you make a whole bunch of it? That being said, the the thermocycler itself, generally I suggest putting the tubes right near the middle as you can. That way the tubes aren't going to have weird temperature differentials. Nothing's going to be too weird. A tube on the outside, it won't be put through different temperatures as the tube on the inside. As a general rule, if you have the space for it, try and keep them as inside as possible. Now, same thing with the thermocycler lid. Tighten that down, because you never know. Some of them do it automatically, especially if they have a spring-loaded top. We have a question, how many student samples can be tested simultaneously? That all depends on the size of your thermocycler and how many tube rack spaces you have. I've seen some with as many as about 72. This one behind me has about 24. It all depends on the thermocycler. So take that into consideration if you're going to run a whole bunch of them. Now, like I said before, this is also a phenomenal time to talk about molecular genetics, how it's synthesized. One of my favorite activities in the lab, is going to sound kind of silly, but we actually acted out how this works. Somebody was a plasmid, he grabbed some DNA. Another guy was a polymerase, he came over, he made everyone stand in a line. It was a great way to learn how it all happens. And it's one of those things where it's almost better to see it in action so you can wrap your mind around it. And my last big tip is, of course, label everything. Once this gets into the tube, it's very easy to lose track of whose sample is whose, what are you supposed to be looking for when you find it. Opening up a big PCR machine of 70 tubes and going, great, where's mine, is an awful feeling. So be sure to label everything. Now, after you have your sample, it's been PCR'd, you're going to move straight into a restriction reaction. The restriction reaction is actually a very, very handy tool, because it allows us to not only differentiate DNA, but it also allows us to, in the long run, do some weird genetic engineering stuff. Because what restriction enzymes do is they recognize very specific regions of DNA and then cut it at that point. So specific that the gene in question we're looking at here today, the mutant genotype of it has a APK1 site right in the middle. Now, APK1 is a restriction enzyme from A. pernix. I believe I have it written down. A. pernix. It is a thermophilic bacteria found outside the Sea of Japan, and it has a very cool restriction enzyme, which, if you have a mutant allele, meaning you can taste the PTC paper, it will cut it in half. So to do that, what we're going to need is a water bath, my elbow shoulder, set to 75. Now, for those of you who have done restriction processes before, you're going to say 75 is a really high number to run a restriction gel or a restriction enzyme at. Well, this came from a bacteria that loves the heat, so of course its enzymes love the heat as well. Water bath set to 75 degrees Celsius. This will allow that enzyme to work at peak function. Now, what we're going to do is we're only going to take a little bit of the sample, about 10 microliters of our sample, and we're going to mix that with 10 microliters of the restriction mix also provided by Geno Sensor. And then we're going to need a bucket of crushed ice just so that this all stays fresh. So, like I said, 10 microliters of the PCR product, which just came out of the PCR machine, and then 10 microliters of the restriction master mix. That master mix is going to contain everything we need, ranging from the restriction enzyme itself all the way to the buffer needed to maintain that reaction. There we go. Now, restriction enzymes are nice because they allow a lot of different things that we can do, and there's a lot of different kits with them included. So, like I said, I'm putting 10 microliters into the PCR mix. I'm going to put that right into the 75-degree water. Now, this will run for about 40 minutes. This allows those enzymes to work on the DNA, activate all the sites that they can, find them and cleave them into. Now, the nice thing about the restriction process is that it can run for as long as you want. I'm very used to running this overnight. The longer you run it, the better it's going to look. Although 40 minutes is about how long it takes to find all those sites. Once it's been restricted, again, great stopping point. You can freeze this at 20 degrees, or negative 20. So there's no need to rush through all of this in one day. You can spread this out over a week or so. Now, after we have our restriction digests all done, we move on to the fun part, which is the gel electrophoresis. Now, gels are very handy in imaging DNA because normally we can't see it. However, when we run a gel, we're able to not only analyze our products. We just had a question come in. How long can it stay frozen? There's nothing in there that will not stay frozen long. What I mean by that is it can stay frozen really indefinitely. What you want to avoid is constantly freezing and then unfreezing it. That will damage the DNA, but if you say do this the beginning of August, and then you don't want to run the gel until halfway through November, it'll stay. It'll stay in the freezer, and there won't be any damage, so long as you're not constantly freezing and unfreezing it. So it'll stay there for a good long time. Now, gel electrophoresis. I'm just going to clear this off because I'm going to bring out the gel. Gel electrophoresis is nice because, like I said, it allows us to not only image a gel, but we can see exactly what we're going to be looking at. Right now, so in terms of this experiment, we have a sample that has been taken from our own cheek cells, which we're looking for very specific DNA sequences. Those sequences, they are in here, but we can't really see them in a tube. So to get this in a form that we can see, sorry, just move stuff out of the way here. We're going to want to use gel electrophoresis. Now to do this, we are going to be using agarose, TBE or TAE. It's a running buffer, both of them. The only difference between those is acid. It's either going to be a borate or acetate solution that you mix with TE, which is a very common supply. We provide it through wards. You can make it yourself. There's no real difference between the two. The only thing you want to keep in mind is whatever you make the gel out of is what you're going to want to run it in. So if you use TBE to make the gel, use TBE to run the gel. Other than that, we're going to need a power supply and a box. Yes.
One question on which is better, your preference between TBE and TBE?
So the question of do I have a preference between TBE and TAE? Whatever one is easier to get. Generally speaking, they're going to run the same. There's going to be no big difference between the two. I've used both. I've been a fan of both. In my mind, I barely distinguish them at all. It is something to keep in mind when you're looking at sourcing the materials, though. If you see it, you're saying, hey, that's B and A is different. What's the deal? There's no big difference. Now, some people say that TBE is better if you're running very small fragments. That's really nitpicky, and I don't necessarily agree with that. So I don't have a preference between the two. Whatever one you have access to is going to be great, just so long as you use that to run the gel and make the gel out of it. All right, so gel box, gel, power supply, and then a couple dyes and a visualizing tool, which I have behind me. So the samples we're going to be running are the samples we have, as well as any of the controls that Geno Sensor has provided, which we'll also have run through that PCR machine. Now, the gel itself is it could be overwhelming to someone who's never had a gel or poured a gel or loaded a gel before. The trick with it is to take your time and know that it's repeatable. There's no big worry if you mess up a gel off the first bat. Only when it comes to loading the gel, you have to be super concerned. The gel in question today is a 2 to 3% agarose gel. What I've done is I've taken 2.5 grams of agarose. I've put it in 100 mils of TBE, and I have put that on heat and dissolved that agarose into solution. So that molten gel is then poured into a box, which is prepared as such. Now, there are different boxes, and there are different ways to prepare it. This one comes with a couple handy rubber stoppers on either end and a comb in the middle. Now, this comb is going to make the divots where we load the gel itself. And it's very important that before you pour it, everything here is in place. If you're missing some of the rubber stoppers, you're going to pour the gel and half of it's going to be on the table. If you're missing a comb, you're going to have a solid slab of agarose. Now, that being said, if that ever messes up, it's very easy to let this cool, pick it up, throw it back into a beaker, and redissolve it. But once you have it like this and it's cooled to a point where you can actually use it, it's very easy to load and then run it. Now, one of the big things that I do recommend with these gels is when it's in molten form, if you have a pre-stain, something like gel red or ethidium bromide, if you're using that in a lab situation, add a couple microliters of that to the gel. That'll actually save you a step down the road. This lets you be able to actually image the gel much earlier than you would if you were using a post-stain. Now, the gel itself is only about 5 millimeters thick, and you don't have to pour it all the way to the top. Now, when you're actually putting the gel into solution here, or into the box, it is a very straightforward way to go. All we do is we pop off these rubber stoppers, and they slide off very easily. And the comb, now the comb is going to be actually stuck in the gel when you first go to get it out. And what you're going to want to do is very gently wiggle it out of the gel. You're not going to want to do it too hard, otherwise you're going to pull some holes. And those holes, any tears in the gel, those will come to haunt you down the road. So I'm just very carefully and very steadily, gently wiggling it, getting some motion, and there we go. All right. So I have my comb out, the gel is all set, the wells are looking good. Very solid. All right. Now what I'm going to do is I'm going to put it right into the gel box itself. I'll move some of this out of your way so you can all see. Now, when you're putting it in here, something that a lot of people forget, and myself included, especially early on, DNA is negatively charged. So it will always try and run to the positive end of whatever gel box you're putting in. Now, DNA runs towards the positive end, meaning that we're going to want to have these wells on the opposite side of our red diode. You can see on the screen there. Now this red diode over on this side, and the gel with the wells on the other side. That's going to go right into the middle of the gel, and some gel boxes are designed a little bit differently, but all operate on the same principle. It goes right in the middle, and then we're going to take our 1X, in this case it's TBE, and we're going to pour it into the wells to make sure that that gel is covered. What I generally like to do is fill one side, and then fill the other, and then very gently get a little bit over the top. And that gel is looking pretty good. All right. So now we're going to load the gel, and I'm going to clear some, and we're actually moving to the camera on here so you can see how we load the gel once I get all the stuff out of the way.
You use a battery-operated gel box?
Yes, battery-operated gels are something provided through wards. They are great for the economically minded teacher because you don't have to get all this stuff. A battery gel will operate with a battery directly attached to the gel box, so you don't have to use a power supply or anything like that. Speaking of power supply, I'll get this guy out of the way. And we will move right over here. All right. So here you see our gel with our beautifully done wells. And what I'm going to do is I'm going to prepare a solution to run through the gel. Now, I have our restricted DNA, and we are going to be taking... so we have a question. How many gels can be run with the thermal cycle or the power supply I'm using today? This power supply has space for 4 gels you can run simultaneously. Other power supplies might have room for 2, some might have room for 1. All depends on the power supply. This particular one can run 4 at the same time. So I have our DNA right here, and I'm going to be taking 20 microliters of it. Remember, this has been cut with the restriction enzyme. So I'm going to be taking 10 microliters of it, and I'll be adding about 2 microliters of loading dye to that. Now, what the loading dye does is, the loading dye, in this case, it is a 10x loading dye offered through wards. This is good because the loading dye will let that gel, or will let the DNA sit in the gel, so it won't go floating off somewhere. All right, get that in there. So we're going to want to prepare the samples of the DNA ladder, which is a ruler of sorts, as well as our controls, the positive, the negative, and then the heterozygous control, so we can see what we're looking at. And we're also going to do this any and all samples we do. 10 microliters of each DNA, and then about 3 or 2 microliters, depending on how much you want to run. I do recommend that you run more than 10 microliters of DNA, especially if this is your first gel. It is much easier to see more DNA than it is less DNA. Now, each gel, depending on how thick you've poured it, can hold upwards of 20 microliters or so. Today, I'm going to be putting about 10 in, simply because I know this DNA is very, very thick, or high concentration. So when you're going to load your gel, this is generally when most people, if you're going to mess up a gel, it's at this stage. Either you're going to lose your DNA into the ether that is the TBE floating around it, or you're going to puncture a hole straight through the gel and you're going to end up loading underneath the gel. Now, this is something that comes with experience. The more gels you do, the more times you load, the more comfortable you're going to be with it. I probably kept a running tally of how many gels I ruined. It's probably somewhere in the teens. So it's something that you're just going to be very careful about. And when you do it, I always find that it's easy to steady the gel with one finger, or steady the pipetter with one finger, and then come in at an angle. That way you're not poking a hole right through it. So very carefully, I'm going to come in and I'm going to load the second gel in. And I'll tell you why in a minute here. So I'm going to come in and just want to make sure I'm coming at the right angle. There, I'm in the gel. Very carefully, I'm going to, there we go. I'm going to unload that. Now this is a pretty thick gel. You can see a very faint blue right in there on that second one in. I poured it pretty thick, but that's about 10 microliters. So remember, the thicker the gel, the more DNA you can fit in there. So I do recommend running more than 10 microliters if this is your first gel. For my purposes here today, 10 microliters is what I'm going to show. So once that's in there, you're going to fill out the rest with a ladder, which is the way we can tell how big those actual fragments are. A gel ladder is going to have pretty regularly spaced intervals of bands, and they will usually come with a with a means of saying how big something is.
We have a question. Teachers don't advance.
So the question is, if you want to prep this in advance, how many gels can you make, and what's the storage like for them? So a agarose gel is a very it's a tough little gel. These are 2% gels, so they are very dense. Physically, they are hardy. If you want to store them for a long period of time, what you do is you put them in a plastic bag, fill that plastic bag with whatever you use to make it, be that TBE or TAE, and then let them sit in the fridge. The only thing that could possibly happen to these is that they dry out, and when they dry out, they are generally just chucked because you can't do anything with them. But to keep them moist or keep them wet and cool, they'll stay for a very long time. Now, what you can also do is you can make a lot of agarose in a beaker solution and then cover that up. That will also stay, and then you can pour it down the line as you need. So that is loading the gel. So I will take this out of here. Samples per kit. So per kit gives you about 20 to 30 samples. I believe it's close to 30 because the folks at Geno Sensor do overpack their kit a little bit. So a standard classroom is about 24, 26 children or students in the classroom. This kit will do about 30. So now we're going to actually run the gel. Now that it's loaded, the hard part is done, so to speak. So I'm going to bring back our gel power supply and get it all set up. Now this gel is going to run at about 100 volts. Now, the DNA we're looking for isn't too big. It's only about 100 to 200 base pairs large. So this is a pretty thick gel, but even so, it's not going to take too long to run this. And you want to be very careful with running gels, especially at this size, because you're going to find that gels separate as they move. That is their point. So smaller bits of DNA will start separating earlier on from larger bits, and then you're going to find that the DNA itself will have separated, and then the small bits are just going to run with it. So it's very easy to find yourself looking at a gel going, where's the bottom half? If all the big bits are halfway through and the small bits are gone. So this is going to run at 100 volts for about half an hour or so. And remember, like I said, run to red. DNA is negatively charged. We're really going to want to make sure we get that right, because nothing is as heartbreaking as looking at a gel run the opposite way. Now we're going to run this to about 2/3 the length, and then setting this thing up is very straightforward depending on your gel box. Some require a top to be a full circuit, so I'm going to pop that on, pop that into place, and then the positive and negative ends of the electrodes go right into the gel box. And then, of course, all all power supplies are a little bit different. Some give you a little manual tuner, others are digital. This one in particular is digital and will allow you to really set how long you want it to run and at what voltage you want it to run. So 100 volts for 30 minutes, Very easy to set up with this. All we're going to do is plug it in to the negative and the positive ends. Now, here's a quick thing that's actually really cool about these. If you do end up putting the gel in backwards, you're saying, oh no, I've already loaded the gel, I've already put it in there, how do I get it out? It's backwards. Well, unfortunately, a circuit is a circuit, and there's nothing too crazy about it. So if you have to switch the diodes, you just do it like so, and then it's running in the right direction. Who have a question, is 100 volts the best practice? Yeah, generally. For this size of DNA, 100 volts is going to be pretty good. It's going to be about 500 or so milliamps. Some people run gels based on voltage. Some people run gels based on amperage. I've always run gels based on the voltage. That's going to be the best bet for just knowing how far it's going to be. Running at 100 volts, that's going to be good. Normally, people run at about 90 because this is a thicker gel, and we're going to look for real good separation on the really big genomic DNA and the small bit we're looking for. We're going to want to run at about 100. So yes, 100 is going to be the most best. All right. So after running the gel, we're going to have to image the gel. Now, imaging the gel is going to be no issue if you've pre-stained, which we do recommend. Now, pre-staining means you have a pre-stain or you have a normal stain which you can use as a pre-stain. And that means you can either image it with a UV light or a ultra bright white light. Very easy. For you, essentially, once the gel's run, you can move on and image it. If you don't have a pre-stain or if you don't have access to one, it's very easy to make, it's very easy to run, it just takes a little bit more time. What you're going to do is you're going to take a 1x stain, or either you're going to dilute it with DH2O, whatever you have to do for the particular stain you have. You're going to take that stain, and after your gel is run, I have a run one right over here, you're going to want to put it in a little container with that stain. Let it stain for, generally it's about 40 minutes, maybe an hour. And what that'll do is that stain will mark where the DNA is, and then you'll be able to see that DNA on whatever medium that stain works with, be it UV, be it light, whatever it is. Now, I have in my hands a gel which I was able to run the other day, so we'll actually be able to see what it's supposed to look like. So reading the gel is very straightforward. You're not going to find too many issues with this, so long as you have a good amount of DNA, PCR worked out well, and you don't accidentally poke a hole in the bottom of the gel. Now, like I said, the bands we're going to be looking for are about 100 or so base pairs in length. So remember, what we'll be looking for is that 100 base pairs, give or take. Now, the homozygous wild type, that's meaning people who cannot taste PTC, we don't expect those to be cut in half because they don't have that restriction site. APE1K will not recognize it and will leave it alone, meaning it's going to be closer, it's about 150 or so. That's a full and complete gene. The mutant type, however, people who can taste it, they will have a cut roughly halfway through it. So you'll see two bands, and as you see on the picture there, the first one in is the latter, the second one in is the homozygous mutant type. They have two bands there. The third one in is that homozygous wild type, no cutting, and the third one is the most interesting one because that has both the gene for the not homozygous, but it is heterozygous. It has both genes, positive and the mutant type. That's some people like me who kind of taste it. Now I'm going to go to this camera over here and actually going to show you a gel that I was able to run the other day. See if I can get this going. Hopefully there we go. All right. Now it's a very faint line right about here, and it looks like the camera's not showing up too well. I'm going to try and maybe do it without the light. Let's get really close here. You can see a very faint blue right there. Now that darker blue, that is actually the homozygous wild type. That wild type came from someone in the audience who can taste and has who cannot taste it. The one next to it, which you can barely see on the actual camera there, that is me. I can taste it a little bit, and if you look closely, you can see roughly about 3 bands. It's very faint, but it's easier with the box. Unfortunately, the bright light with the camera is throwing it off a little bit. And then the first one in right here, that is a gentleman from the office who had a hysterical reaction to the PTC paper, had to wash his mouth off for about 5 minutes after. So, and that is, what we expected after tasting the PTC paper. So after reading the gel, it's fun to see, were your results what you expected? Did you see any weird results? And keep in mind, we are working with genetics here. Nothing's a given. And if you have a student who swears they can taste it and tests negative for it, it happens. But generally, you're going to find about 60 to 70% of people are very accurate with this test. Nothing crazy, so it's not like we're going to be changing anything too much, and you're going to be able to see the results you want off these. Get this out of the way.
It looks as if the gel was run for only a short amount of time.
Yeah. So this gel is a 2.5% gel. You're going to want, because remember, these bands are very small, but a sufficiently thick gel, it will move slower. Now, I was really paranoid about having this run too far. I did not want to have it run off the gel because I have done that a million times and I didn't want it. The dye I used was a blue-orange dye, and it has 2 dyes in it. The yellow dye in it moved all the way to the end. Once it moved to the end, I said, I don't want to risk anymore, so I shut it off. So no, it didn't run too far, but it did run far enough to see the separation of those genes. Generally, when you run a gel, you're going to want to run it to about 2/3 the length of the gel. That way you have plenty of time for separation, but not too far so that you're going to risk losing the bottom end of things. And again, that's why we run a thicker gel for this, as opposed to a 1% or 0.5% gel. Those would be for something if we're looking at bigger pieces of DNA.
Thank you.
All right, so like I said on gel electrophoresis, it can be harrowing if you've never done it before, and it's a very stressful thing to do when you first start doing it. The biggest tip you could possibly take is take your time with it. If you feel like you're going to poke a hole, take a step back, come at it at a different angle, figure out what's going to work best for you. There's a billion ways to load it. There's a billion ways to run it. As long as you follow the basic rules, whatever works for you is going to be the one I recommend. Now, that being said, take your time. Also, record everything. There's no writing on a gel once you've put it in. So if you put something in the gel and you start running it and you don't know whose sample it is, make sure you have it on a piece of paper. Now, I did just have a question come in saying, what size wells should you use? It depends on the person. Generally speaking, I like using the bigger wells. All combs come with 2 sizes. A bigger set of wells on one side and a smaller set of wells on the other. Those bigger set, if this is your first time running it, it is much easier to load the big set. It also holds more DNA. So if you wanted to load 20 microliters of DNA, use the big set. Plus, it's easier to find the holes. Now, if this is something you've done a lot and you want to run a whole bunch of samples on the same gel and you only need about 10 microliters a sample, the smaller well size is good for you because it'll be able to hold more samples. If you have a large class and you want to do something, if you want to combine classes, that's a good way to do it. But ultimately, there's no strong difference between the two. All depends on what you're comfortable with working with. So, like I said, the prestaining this, highly recommended. It kills about an hour worth of time. It is much more fun than, especially if you have a class setting, coming in and saying, gels are done, now you have to wait 40 minutes because your class is going to be over by then. It does give you an opportunity to talk about other stuff, but generally speaking, that pre-stain is nice. Cuts down a lot of time. Now, like earlier, the question as well. Remember, these gels can be saved. If you want to save a gel for later, if you want to save a gel, like an unrun gel or even a run gel, you can save it. If it's run, don't put it in TAE or TBE. Put it in water. The TAE will leak out otherwise. I'm sorry, the DNA will leak into the TAE otherwise. So just have a question come through saying, does Ward's sell DNA synthetic samples of the wild type, heterozygous, or the mutant type to use in case the DNA doesn't work for the students? The kit for Geno Sensor does include those controls. I didn't bring them out simply because it's much easier to load one at a time. So they do include those controls, and you'll be able to see them on the gel when you run them. So if ever there's a question on if something is positive or negative, running those controls with the gel will show you for a fact that yes, that is the case. So I have another question coming through saying, I'm sorry?
It's a comment.
Oh, sorry, a comment coming through saying somebody has those the gel models and they purchase them through Wards. I let the students practice on these before they load the actual gels. It works well. That's great, especially if students can practice beforehand. Letting them practice on on a fake real gel is a bonus. My first time loading a gel, it was with an actual sample, and I poked a hole right through it. And that took me about an hour to get that sample, and yeah, I'm a little bit bitter. So any chance you get to practice, take it. All right. Now, the other cool thing, and speaking of that loading the gel, they do sell, Wards as well, Ward sells special gel loading tips. Now, I am a purist, and I like my tips, but if you are using them in a high school setting or students the first time loading a gel, look at those gel loading tips because they are useful, especially if you don't want to puncture a hole right through the bottom. They're really nice, and they're good for getting a feel of how to load a gel. So this was a very basic kind of foray into the world of molecular genetics. We've touched on a lot of the good stuff: PCR, gel electrophoresis, restriction enzymes. All of those are the bread and butter of a molecular geneticist's work. Genetic engineering, anything with DNA, these are the ones which are going to be used most. That being said, there's a lot of other stuff you can talk about. Restriction modification. Restriction enzymes have a cool cousin called modification enzymes. They actually do the exact opposite thing. Well, not the exact opposite, that's ligation, but they defend against restriction, kind of like a shield. And, if you are halfway through this and you want to expand on that, there's a lot of good opportunities to talk about modification and restriction during the class. Same thing with DNA synthesis and repair. The PCR itself is a very, very small portion of how actual DNA is synthesized. So if you want to talk about the full process, it's another great opportunity. And like I mentioned earlier, Kary Mullis is a weird guy. And that's me saying that. So if you get the chance, look him up because he invented PCR essentially on a dare. And PCR itself is a very informative topic. So if you get the chance, it's not bad to talk about it every now and then. Other kits that are great that do a lot of similar things, DNA fingerprinting provided by Geno Sensor. That is a great opportunity to use more restriction enzymes. It comes in 2 flavors. The flavor of the number on there is a kit that uses restriction enzymes on gels. Great tool to learn more gels, great tool to work with restriction enzymes. And then, of course, our own Wards kit is a transformation kit. If your students really like seeing practical uses for DNA, other synthesis tools, that's a good one because you're actually able to transform pGlow into E. coli. A great opportunity to teach about how bacterial genetics work, great opportunity to talk about the transformation process, how it's useful. really a productive way to kind of get hands-on experience with some of these molecular processes. And then all the materials here, I've put them on the page there. The genetic taste kit, I have the number on there, as well as any of the other accoutrements. The gel box and the power supply. There's a question about the battery-operated ones. Check out wardside.com. There's a ton of stuff there, and if you see something you like, check it out. And of course, if you have any technical questions, feel free to email me. My email's right there. It's austin.osip@VWR. If you need a copy of the presentation, email ScienceHelp, that's all one word, at VWR.com. So if you have any questions, feel free to unmute your phone, ask away. Remember, unmute, it's *6. If you have a cell phone, it is just unmute. Or, of course, feel free to type into the web browser as well.
Do the items that you showed us today come frozen or cold?
Yeah,
I'm a
so...
little concerned about delivery for that.
Sure, yeah. So the items in this particular kit come in two different packages. One is a room temperature package, which contains the PTC paper, the the cotton swabs, as well as solution A and B. Now, the other part of the kit is the frozen section, which contains all the enzymes, all of the different controls, the DNA. Now, that should stay in the freezer. Now, the other question saying when you get this kit, one of the nice things about Wards and what we like to help you out with is it's very annoying to, especially if you do a lot of your ordering in the summer, to get a shipment in the middle of July, have it sit on a loading dock or something for a weekend, and all your DNA is denatured. No one likes that, and it doesn't really help you out at all. So actually, what we offer is if you want to get this kit in August, you can order it in July, and if you know of a Wednesday, Thursday, or a Friday that you're available to actually get this kit, you're available to pick it up from your loading dock, put down that date, and you can order it in July, get it in August, and that way you're not running the risk of a 80-degree weekend for DNA, which is awful. DNA is a tough molecule. It's not that tough. So again, if there's any other questions I can answer, *6 to unmute your phones, or feel free to type into the web chat any questions you have.
You're very personable, and thanks for the great presentation.
Thank you. Again, thanks everybody for tuning in and seeing the webinar. It's great to actually have some interest in all this stuff, because it is a great opportunity to teach a lot of cool stuff about molecular genetics, anything like that. We're able to really teach a lot with this kit.
Austin?
Yes?
Oh, hi, this is Holly. I'm in Iowa. I have a question.
Hi.
I'm not even sure for this, but I have a VWR inverted microscope, and I'm going to be doing a lot of gel electrophoresis, PCR stuff, but I don't know what type of imager or camera to get. Would you know that too?
Sure. Well, the imager all depends on what you want to use for that staining dye. The question for people who may not have heard is, what imager should I use to image anything? It all depends on the dye you use. For example, a very popular one is gel red. That is one that can be used under a UV dye. It intercalates the DNA and it works a lot like ethidium bromide. It'll actually fluoresce under UV. The one I used is a one provided through Wards. You run it with the DNA, you stain it afterwards or before, and then the DNA will turn blue and it will just show under normal light. So any imaging, it all depends on what you want to stain with.
Okay, and I guess the hard part for me is I don't really know exactly the project I need to, maybe I need to wait on that a little bit.
Okay.
Too.
Well, if you have any
do a
questions, that in particular, feel free to email sciencehelp@VWR. We have a great support staff there, and if you want to know anything in particular, email them, and then we'll work with you to get that solved.
Okay, great. Thank you so much.
All right, thank you.
Bye.
Thanks for the informative presentation incorporating PCR and gel electrophoresis with a PTC genetic test.
All right, yeah. So this is really awesome because very rarely do you get to see a genotype and phenotype actually interact. I can't tell you how many genetics courses I've sat through, and it's been, oh, did you know that X, the blue eyes are a mutant gene? And I go, oh, cool. And it ends at that. It's really nice to see a kit that lets you utilize, hey, there's this about you. And hey, guess what? This is why that is. And hey, guess what? Now you can see it. You walk away going, oh, that's really cool. So the folks at Geno Sensor, I have to give a shout out to them. They are really helpful, and they've designed a very strong kit. All right. So I think we're going to wrap it up then if there's no other questions. If you do have one down the road, like I said, Science Help or email me, and we will, I will start picking up.